Everyone. Thank you for joining us today. We have two folks joining us to present, and today is our final Northeast Extension Fruit Consortium seminar for the season. Thank you all for joining us. Those of you who have been with us over the past several sessions, thanks for taking the time and supporting us.
Thanks for having me, everybody. I hope everybody's having a good week so far. I'm going to talk to you about some of my work looking at cold hardiness, primarily in apple rootstocks. And it's sort of framed in trying to understand how cold damage might be impacting apple tree decline. And so I'll just get started right off the bat with some photos of declining apple trees with a specific type of decline that I call rootstock shank decline. And so lots of different things can kill apple trees. But when I started at Cornell in 2022, I came from a background of cold hardiness and grape. And so I was pointed towards this problem in apples because of the sort of correlation between declining apple trees and instances of cold weather. And so when we go out to the field and we look at the trees that are suffering from rootstock shank decline, they look like other forms of apple decline. So pictured here on the left, you can see this tree is like a little bit more chlorotic than the trees behind. This is really evident in the center picture. And then if you track trees long enough, you eventually get to complete collapse of the trees or dead trees in the orchard. When we look, this apple decline can happen to young trees all the way up into trees around maybe 13 to 17 years old. We don't typically see it in very old trees. The young trees tend to show compromised bark with discoloration, along with decreased vigor and leaf color changes. And so pictured here is an example of a gala that is in decline. You can see the enhanced anthocyanins in the red leaves. In the center panel here is the partial chlorosis that Honeycrisp does naturally. But when you see a declining Honeycrisp, you can see the chlorosis is really enhanced and the leaves actually get curled and bubbly and a little bit of necrotic tissue along the leaf margins. With trees that are showing weakness, if we split them in half with a bandsaw and we look at young trees, here's Gala on G11, and next to that is a picture of Pink Lady on EMLA 9. What we see is that there is a specific type of damage that's affecting the vascular tissue. So here you could see that this is the graft union between the scion and the rootstock. You could see the sort of yellow alive vasculature right at the graft, and then this zone that's sort of necrotic. And then as you get towards the root, you see that live tissue comes back. And you see the same thing over here on the older tree with no real obvious graft necrosis going on. So this looks like something that's affecting the vascular tissue of the rootstock shank. These young trees often have very healthy roots and lots of suckers, suggesting that the problem is not coming from below the soil, but it's between the soil and the graft itself, so the rootstock shank. And you can see some of that pictured here. When we look at a little bit older trees, we also see the leaf color change and a vigor difference in these trees. But if you go down and look at the rootstock shank, you get really extensive bark damage. And pictured here on the left is an example. You could see that the bark tissue does not look particularly healthy. And if you pick around at that bark with a knife or with your hands, you'll see that it's mostly hollow, that there's this thick outer bark layer that if you strip off, you get this intact rootstock shank, but really no vascular connection between the scion and the roots. Often underneath these chunks of bark, there's a lot of biotic pathogens or pests and pathogens, a lot of fungi, a lot of slugs. There's often a lot of sawdust from beetles and other sort of borers. And so the tree is very compromised, and it seems to be that this rootstock shank is a point of weakness. I want to contrast that sort of bark flaking and damage to healthy bark cracking. So a lot of the Geneva rootstocks have this bark sloughing phenotype where you'll get these very thin bark layers will peel off, and you'll see this underneath this amber colored tissue. If you wrap on that with your hand or with a knife, you'll see this very solid, very secure tissue. It's perfectly healthy. What I'm really talking about is when you get these deep fissures and dark wood underneath, that when you pull away, you could see that structurally the tree is sound, but vascular tissue has been completely eroded. Here you can even see there's a lot of moisture underneath this tissue. So it's really conducive area to other biotic organisms. On trees that you don't find the complete girdling of the shank, you can find partial girdling, and this animation shows you an example of a tree in eastern Washington or eastern Massachusetts. Here you can see the scion is here connected through this little strip of vascular tissue that goes across the graft union down into the soil. And so what I'm trying to illustrate here is that all of the water needs for the canopy of this tree has to go through that little attached strip, and all of the sugar going to feed the roots is also going through that detached strip. And so you're really looking at a very compromised ability to move water and sugars up and down the tree. And so this leads us to the question, what could cause damage specifically to the rootstock shank? It doesn't kill the roots and it doesn't kill the scion. And so that's where the cold hardiness aspect of this comes in. We see that there's a correlation between episodes of tree decline with winter exposure. And specifically, I've been told that large swings in temperature are thought to be a bigger problem than consistently cold winters. And so the basic hypothesis here is that acute cold acute cold damages the tree, that weakens them and sets them up for future decline. But in order for this to actually be a mechanism, we need to see that rootstocks and scions differ in their cold hardiness throughout winter. So that's the approach to understanding this phenomenon. I want to spend a little bit of time explaining what cold hardiness is and how it manifests through the winter. And so cold hardiness is simply the ability of a tissue to survive all of the minimum temperatures through winter without sustaining any damage. And pictured here on the right is in a kind of a cartoon representation of that. In red and blue are the highs and lows of a given winter, and this dashed line here would be a cold hardiness curve, where we're tracking a changing ability of the tree to survive freezing damage. It starts at the beginning of winter with this phase where they gain more and more defenses. This is the acclimation phase. In the middle of winter, they have maximum hardiness, and it's sort of a maintenance phase. And then as temperatures rise in the late winter, early spring, the trees will rapidly lose that defense as they get ready to grow and break bud. Important to this whole process is that the tree needs to go dormant. If you delay dormancy in the fall, you interrupt the process of acclimation, and you can't gain maximum hardiness. And so at the beginning of winter, the trees are in a state of endodormancy, where they are very resistant to heat and are able to gain cold hardiness if you give them low temperatures. As we go through winter, the trees are exposed to chilling hours, which is temperatures between 32 and 50 degrees Fahrenheit, depending on which chill model you use. And the idea here is that the trees track the length of winter by adding up all of the hours they spend within that temperature range between 32 and 50 F. For apples, it usually takes around 1,000 to 1,200 hours before they transition from endodormancy into late winter dormancy or ecodormancy. And the defining characteristic here is that when you're ecodormant, the trees are not growing because the temperatures are cold, but if they were to be exposed to heat, they would rapidly lose their defenses in anticipation of spring. So the chilling hour in the Northeast typically lands right around January 1st. It moves from year to year depending on how many chilling hours occur. But if you take an average over time, January 1st is a good sort of reference point for you to think in your orchards. Warm temperatures before January 1st are not as much of a concern from the point of deacclimation because the trees are endodormant and they're resistant to heat. But after January 1st, they're very responsive to heat. Now, of course, when we talk about grafted apple trees, we're really talking about two different genetic individuals. The rootstock is grafted to the scion. And if you think about cold hardiness, this means you can have different cold hardiness curves depending on the rootstock scion pairs. Another thing to consider is that the rootstock shank and the roots themselves can often be covered with snow. And because snow is a very good insulator, the tissue below the snow layer can experience a very mild winter condition. So imagine instead of these low temperatures of midwinter down here below -4, they're actually much warmer around 14F because that snow is such a good insulator. The consequence of this is that the rootstock cold hardiness curve may be much less cold hardy than the scion if you have instances of really heavy snow, particularly if it's heavy snow in the beginning of the season that then melts by the end of the season. And so it's really important to understand the capacity of rootstocks and scions to survive freezing in response to these different forces so that we can adequately pick combinations as our winters change. And so here's again that picture of the pink lady on MLA9, showing you the different types of damage. The model for how all this works together is that freeze damage occurs, it damages the rootstock shank. The cambium tissue tries to repair that vascular damage. While it's repairing, it's also emitting volatiles that are attractants for insects and fungi. And these pests and pathogens can attack these weakened trees, which creates a feedback loop, resulting in more and more damage. The consequence of all of this is a decreased root function by reducing the amount of sugar that gets to the roots. We have other compounding factors here. The dwarfing rootstocks we grow are small root systems to begin with. We plant them in high density, so they're in high competition. And we have other stresses that impact root function added in. All of this together, while the canopy is developing and increasing its water demand due to high crop load and then heat and drought conditions in the atmosphere, all leads to an imbalance between what the scion needs in water and what the roots are able to provide. And this, the theory is that this leads to eventual decline of the trees. Okay, so now let's talk about how we measure cold hardiness. So there's a couple different methods. One method is oxidative browning, and the idea here is you freeze the plant tissue to lots of different test temperatures, and then put them at room temperature and see how brown they turn. And we use the level of brown as a relative measure of damage. And so what that experiment looks like, if you look at cross sections of rootstocks, is something like this. So here I'm showing you multiple different freeze temperatures, 3 different rootstocks, and then how they go from green if they're not frozen, or purple in the case of B9, all the way down to watery straw color, dark brown, or in the case of B9, again, sort of a faded purple. And what you can see here is when you look across this is you can see a gradual change from green towards brown or green towards straw, but that the browning pattern is not always super consistent. Here we can see there's a ring of brown. Here you can see it's a little straw colored. Here you can see that there's a lot of purple fading, but then not much purple fading at this colder temperature. And so the phenotypic scale using color can be a little muddy. So we use another method. Oh, I forgot the slide was in here. Sorry. One of the other complications here is that we're also looking at multiple tissues. And so here we're looking at how brown is the xylem turning, how is the phloem turning brown, and how is the cambium being affected. And so all of these different tissues could have different freeze cold hardiness points. And so looking at it across all these tissues makes it a complicated situation. The other method we use is electrolyte leakage. This uses the same concept. We collect a bunch of little stem segments, we freeze them in tubes at lots of different temperatures, and then we measure their conductivity of the water that they're in using a electroconductivity meter. In this case, it's attached to a robot. And what we're basically doing is we measure the leakage at different test freezes. So in this case, at -5, -15, -30. And then we measure that leakage again after we freeze them in a -70 freezer to really make sure we kill them. And then you get a relative damage. And so just for example, here the EC at the first freeze is 25 and the final is 100. So we have 25% damage. So it's just a way of damage across different treatments. So when we look at that data, it looks like something like this. So here is freeze temperatures going from the freezing point down to -76 Fahrenheit, and then percent damage from 0 to 100 on the Y scale. Each one of these little dots is one of our electrolyte leakage measurements. And then we plot a dose response curve in order to compare the curves at different times. And so here you can see November's curve in dark blue. December's curve is shifted towards cold, so it gained cold hardiness. And then January and February, it shifted back to the right, so it lost cold hardiness. When we look at how these overlay between browning and electrolyte leakage, we see there's very good correlation in general between the low levels of damage in green tissue versus the high level of damage in brown tissue. And we just pick the LT25 as a reference point because it's the point where we get brown cambium and xylem tissue, and we use this measurement to compare different rootstocks throughout the different winter time points. So I'm going to show you data for the last 3 winters, not counting our current winter because we're still collecting data. And what I wanted to show you is the highs and lows of those winters over the last 3 years. You can see how different the last 3 winters have been. They fall into sort of different characters. The first winter, 2021 to 2022, was a cold U-shaped winter. The 2022 to 2023 was a false spring winter where we were decreasing in temperatures, but then we had a big heat up right after Christmas, and then another freeze in February. And then last winter, we had a U shape, but it was much more mild than the first year here. So a mild U-shaped winter. And just for reference, that chilling hour time point that I talked about in the beginning, the point where we want to look at the difference between endodormancy and ecodormancy, you can see that that window changes size based on the winter and also the timing of it based on the winter. And so we transitioned between these 2 phases, this heat sensitivity phase, during a period of cooling temperatures in the first year, warming temperatures in the second, and then cooling temperatures in the third. And that's important when you look at the curves a little bit later. All right. So let's look at that first year. So this is the cold U winter. And what I'm showing you here is the LT25 curves for Honeycrisp and Gala, and then B9, M9, G11 on the top, G214, 935, 969, G41, and G257 on the bottom. The dashed line is the freezing point. The dotted line here is the cold hardiness of M9 in the middle of winter, just as a reference point. And so what you can see here is in the U-shaped winter, all of the different selections that I'm showing you here were very cold hardy. At no point does the minimum temperature of the winter intersect with the U of cold hardiness. So they were all safe in the cold year. If you compare using M9 sort of as our weakest rootstock, you can see that the scions and B9 are sort of as weak as M9, but the G series rootstocks are all more cold hardy. So everybody's good at cold hardiness, but the G series in this particular case are more cold hardy than M9 and B9 and the scions. Now, when we look at the fall spring winter, things are very different. So here again, here's the minimum temperatures. You can see that big warm up in the middle of winter. And what you see is not a U shape for most of these cold hardiness curves, but this sort of weird zipper or W shape. And what we're seeing here is that in a mild winter with a false spring, we see a lot of deacclimation or loss of cold hardiness in our rootstock samples in particular. So we had good cold hardiness, but then they lost a lot of that defense in response to this warm up. And as a consequence, B9, M9, G11, 935, 969, all of them lost enough cold hardiness that we predict that there was at least some damage in the field. I should mention that all of these samples are collected from the field here in Geneva, either from rootstock liner beds or from Terrence Robinson's got a scion block where I click the scion wood. So basically the take home message here is that in a mild year with a false spring, we see really rapid deacclimation in a lot of the rootstocks, almost all of them. And in this case of this winter, that put these different rootstocks at risk of freeze damage. Now in the third winter, this was the mild U winter. Here you can see the return of that U shape response for all the different samples. And what you see here when you use the M9 comparison point is that our scions, B9, M9, G11, and G214 are all weaker in the mild year than they were in the cold year. 935, 969, 41, and 257 all retained very good deep cold hardiness in the mild year. So as long as the shape of the year was still U-shaped, we got a really good response with this bottom set of rootstocks. And that's just basically what the text here says. Everybody gained enough cold hardiness to avoid damage, but we start seeing that some of the G rootstocks are better than others. And then the final year of data is our current year, which I didn't show you what the shape of winter. We're still collecting data. This is for the first half. What you see here is very good deacclimation, or sorry, very good acclimation processes in all the different samples, but none of them are really getting super cold hardy. Again, this is the M9, the weakest rootstock in the first year, sort of its level of cold hardiness. So good acclimation, but weak overall. If we overlay the cold hardiness of all years together, what we start seeing is a pattern amongst these rootstocks. And so again, here's Honeycrisp and Gala. What we see is that there is a series of lazy rootstocks. So here you can see B9, M9, G11, and 214. While they all attained really good cold hardiness in the blue year, which is the coldest year, the other 3 years, they've been very weak. 935 and 969 do a little bit better, where they have deeper cold hardiness. Here you can see 935 in the purple, and the orange years are just as good as the blue year. Bad in that warm fall spring. 969 this year is also looking very weak, but in 2 of the 4 it did okay. And then in G41 and G257, we see good cold hardiness in all 4 years. And so we're starting to see segregation of the different rootstocks as they relate to cold years and warm years. Now, I don't screen just these different rootstocks and these 2 scions. I actually do 22 rootstocks and 22 scions. But in an effort to not be too overwhelming today, I just wanted to highlight these. I did want to share this as a preliminary summary of different rootstocks and how they've performed in the cold winters versus the mild winters as sort of a rough gauge of what to think about as you're planning new orchards in your regions. If you believe that winters are getting warmer, which they are, and you want to know which varieties are sort of the most resilient to those warm winters, then you want to look at this mild winter section and you want to pick from the moderate or hardy category. And so here you see that in the samples that I've examined, 257 and 41 are the most cold hardy in all years. And then you have a series of these other varieties which are a little safer bets in mild winters. Again, in cold winters, all of these different genotypes are fine. It's only in the mild winters that we're starting to see a potential problem. This is not my direct recommendation because we're still working on the data, but I wanted to share that with everybody. The other thing I wanted to show is that depending on the winter timing and the combinations, you can have differences in cold hardiness in scions and rootstocks. And so here in 2021, 2022, if we look at Honeycrisp as the scion, G11, which is weak, and G41, which is hardy, you see they all have a good U shape, but the scion is the weakest of the 3. In the fall spring year, you can see that the scions are more cold hardy than G11, but not G41. Again, in the mild winter, you can see here in late winter, they're more cold hardy than G11. And then in the current year, you see they're all sort of tracking together. So that concept of time points of winter where the scion may be more cold hardy than the rootstock or vice versa, what we've demonstrated is that, in fact, yes, depending on the winter, you can have rootstocks be weaker than scions at different points in winter. So to summarize that, there does appear to be the necessary variation in rootstock cold hardiness, particularly in warm winters, to explain why some trees may be taking freeze damage and some trees may not. B9, M9, G214, and G11 are all on the sensitive side. G41 and G257 are the most resistant from what I've screened. I didn't show you a lot on scions. They tend to have hardiness similar to B9 and M9, but they are much less responsive to warm temperatures. So in this particular case, the scions are not the weak point in the grafted tree. So next I wanted to address whether or not we can recreate the decline symptoms that we see in the field with freeze damage. So this is all real preliminary. It's mostly looking at some cool pictures. But what we do is we're freezing whole trees now instead of segments. And so we take, in this case, the picture on the left here is G41 and G11 rootstock liners. We plant them in a trench in the field so they get a normal fall and normal winter, and then we pull them from the field. we put them into these insulated root boxes to protect the roots, and then we freeze the whole top of the tree to lots of different temperatures. And so pictured on the right here, you can see the comparison between gene 11 and G41. When we freeze the whole tree at -4, -13, and -22, and we look at browning of that tree, you see that same pattern I showed you before. But if you put these in the greenhouse and then see whether or not they're able to grow, we see that that browning data and the regrowth data actually broadly correlates. And so here you see G11 at -4F, the trees grow fine after we put them in the greenhouse. At -13F, we kill the top of the tree entirely. G41 at -13F, the same temperature that killed G11, the trees are fine. But at -22F, we've killed the trees. And so we see that these assays, browning and electrolyte leakage, they broadly resemble what also happens in tree regrowth. More importantly, or cooler in my book, is the trees that survive these treatments, after several months of growing in the greenhouse, we noticed that several of them started to collapse. And so here's a couple of pictures of those rootstock liners flushing out and growing. And then over time, they started to collapse from the tip of the tree down towards the base. And when we look real close, we can see here you can see the collapse zone in this little V-shaped interface, is that the top of the tree is collapsing in a downward motion. And as this sort of collapse zone passes the growing tips themselves shrivel up and die. You can see another example over here on the right. When we destructively harvest these trees, what we see is above the collapse zone is that the xylem and the phloem is all dried out and brown, but then the xylem damage is extending down the trunk of the tree from the inside out. And what we see when we do a number of these different cuts is that that xylem damage seems to be a precursor for the collapse of the phloem at the outside of the tree. And so what I think is happening here is the tree, we've killed some of that inside xylem tissue. As it's drying out, it's structurally collapsing the phloem and the cambium on top of it, and the whole tree is collapsing in a downward motion. Here's another example from the tip that's starting to collapse. Here's the picture of the foliage moving down this dissected tree. And then this is a zoom in. You can see that there's some brown streaking down the pith all the way towards the bottom of the tree. This part of the tree was protected with foam insulation. And you can see that that brown cambium and phloem layer all along the outside there. And then once you get down to the protection, it's also green. And so what we're seeing here is a general collapse of the tree. It looks like it's dying sort of from the inside out. And that this broadly reflects the same sort of senescence pattern we see in collapsing trees in the field. So we moved from killing intact trees to doing this with grafted trees, because ultimately we want to see what happens when we have this sort of dance of the rootstock and the scion. And so here's an example of some data that we actually just pulled a couple weeks ago. This is a proof of concept. So what we have here is we have grafted trees. We freeze them in the exact same manner as I described before. We put them in the field, we put them in the root box, we freeze them at lots of different temperatures. What you're seeing here is a Honeycrisp. This is grafted to the rootstock G214. And what you're seeing here in this sort of yellowish streak is I've taken my pocket knife and I've cut just a thin layer below the bark from the scion across the graft all the way down to the base of the tree. At the base of the tree, we include foam insulation to protect the roots. And what I've boxed in here in black is the graft zone between these two pieces. And so here you can see the rootstock of G214, when you cut it, is slightly browner than the scion, just naturally. At -4, both parts of this tree are alive. But when we look at -13F, these are whip and tongue grafts, and this is turned on its side. So what you're seeing here is the brown tissue of the rootstock. At the base, the vascular tissue is protected by the foam and it's alive. The scion is alive, but as soon as you hit that rootstock tissue in the shank that's exposed to -13F, we've killed it. When we look at -22, the same situation. The scion is alive, the rootstock has taken damage, the foam has protected the base, the roots are still alive. And then at -31, we've reached a temperature that now has killed Honeycrisp as well as the rootstock, and you no longer see the differentiation at the graft. And so what we've shown here is that with this particular combination, that an exposure at negative 13F or negative 22F causes damage to the rootstock specifically and not the scion, and that this may have impacts on the way that this tree is able to survive once it starts to regrow. Here's another example of what we've seen. So these, again, this is a Honeycrisp on B10. In this case, we froze at lots of different temperatures. When we look at the survival at negative 4F, 10 out of 10 trees survive. At 13F, 6 out of 10 survive. And at 22F, 2 out of 10 survive. But when we look at the trees that survive at negative 13F versus 22F, and we put them in the greenhouse, they start growing perfectly fine. We cut them. You can see the oxidative damage at the rootstock. The shoot's still alive. It's growing, but now we're starting to see decline of these trees, particularly when we have high light days in the greenhouse. So basically when photosynthesis turns up, these trees are calling for more water from their roots and they're not able to get it. So while we haven't had this tree fully collapse yet in the greenhouse, what we're seeing is that these freeze damaged trees, they're not completely dead. They may be walking dead. This sublethal or soon to be lethal damage is contributing to weaker trees that are in particular sensitive to low water availability. And this suggests that there's a potentially important aspect to mitigating freeze damage in that irrigating trees after potential freeze damage may be a smart move because these trees are going to automatically react as if they're being drought stressed. This is hot off the presses. I'm still working on this experiment, and we'll be putting out some extension work on this, and we can certainly send it out to anybody who's interested. So keep in touch. What we've managed to do with this experiment, though, is demonstrate the concept of freeze damage to the rootstock can occur at temperatures that don't damage the scion, and that can have lasting effects on the tree growth. All right, the final thing I'd like to share with you is that we are trying to move from doing these experiments here in the field to identifying declining trees at the orchard scale using some drone technology. So we're using this Mavic 3 drone with a multispectral camera, and we're flying a couple orchards up here in New York, trying to quantify declining trees in the orchard to get a better idea of the timing of decline and the percentage of decline. Here's just some imagery of what the drone is able to capture. We can very clearly see spots in the orchard where trees are in decline or have already died. We're using different vegetation indices, basically how green are the leaves, to identify trees that look like they might be weak. We're also trying to characterize vigor. What we see is that we see a very big difference in the way that different cultivars are imaged, and that greenness is cultivar specific. In that here you can see the distribution of how green a leaf would be on Fuji, Gala, and Pink Lady. You can see Fuji and Pink Lady are in general more yellow than Gala, so this makes it a little complicated to use drones to identify weak trees, but we are using this to try to parse through different tree blocks. We are using images of vigor based on how fast the canopy is closing. So here you can see a really good example of a low vigor tree in amongst the other healthy trees in the orchard. And so using these different metrics, we can start to identify the rate of declining trees in a given orchard. And the whole idea here is to work to identify trees that are weak and then possibly intervene on their behalf to head them off before they reach full decline. So this is a work that's in process. I also wanted to mention that we're also trying to identify this stuff with hyperspectral imaging, which is looking at spectra of light outside of what we can see with our eyes. So we use these handheld cameras, or we use these computer-driven leaf punch assays to try to understand which wavelengths of light could be predictive of damaged trees. And so here's just a couple examples. One thing that we've learned from this is that there are specific wavelengths of light that are associated with tree decline. So here is an example of UV light into infrared light, the nanometers of light that define those different wavelengths. We see in the visible light here from purple to red. And so what we're doing with this hyperspectral approach is seeing if there's any other wavelengths of light that could be used by drones to identify the weak trees. And what we see here is based on how high the black line goes, so from 0 to 1.5, this tells you how important that wavelength is at identifying a weak tree. You can see that there's this little blip here in the green zone. So this is what I was showing you before. How green the leaf is a good predictor of whether it's a weak tree. That's not perhaps all that helpful because we can see that with our eyes. What is really interesting is as you go into the infrared, you start seeing these large peaks, and these peaks are associated with the water bands or leaf water status. And what that's telling us is that the leaves on declining trees in the orchard are under water stress. So this is also backing up what I just showed you with the freeze damaged trees, is that they tend to respond as if they're drought stressed. And so that suggests that we could actually pick out declining trees, perhaps using thermal imagery better than hyperspectral imagery, because those trees should glow as if they are overheating on hot days. And then finally, there's some other peaks here, and these peaks are associated with different metabolites, different starches, different fats, different proteins. And so we're working to identify if any of those could be used as a biomarker. And with that, I'll just wrap up. So I've heard a lot of stuff, but the take-home points are, in a cold year, there is variation in cold hardiness, but all of our signs and all our rootstocks that are currently in use are perfectly capable of handling a normal cold year. Scions are less of a problem in general because their temperature responsiveness to warm temperatures is muted relative to rootstocks. In a mild year is when we start having some issues. Some rootstocks are demonstrating reduced acclimation and enhanced deacclimation, or the loss of cold hardiness, when we have mild winters. In particular, M9 and G11 and even G214 look like they're problematic. It is important to note that cold hardiness is just one of the traits that you need to consider when you're choosing your rootstock. Depending on the way that our winters change over time, that trait itself may become more and more important. So we do have to keep it in context that cold hardiness has to be balanced against other traits. We were able to document that there is variation between scions and rootstocks and cold response. We are able to document that trees decline after suffering sublethal freeze damage, and we're able to document that the rootstock and the scion can be differentially sensitive, and that leads to weakened trees. And so now our work is starting to shift from characterizing cold hardiness more towards testing to see if any products can enhance cold hardiness or any products can enhance repair after freeze damage. And with that, I would just say thank you for giving me the space to share this with you today. My email address is there. Please feel free to contact me with any questions. The crew that helps me out comes from my lab, Gennaro Fazio's lab, Terrence Robinson's lab. Everybody's up here who contributes to this. And we've been funded by New York State ARDP funds, Hatch funds, and none of this work could be done alone.
Welcome, everyone. I'm Renae Moran, and I work at the University of Maine. And I've been doing a lot of research on peaches in the last several years. I do know that growing peaches does come with a high risk for repeated yield loss because peaches aren't fully hardy in the northern parts of the region. But I'm aware of growers who actually want to plant peaches. So I've been testing the hardiness in a lot of the different varieties that are fairly new to find out which of these varieties we shouldn't be planting and which we should. And even when I do know we shouldn't be planting some of them are just so good that I have to have them in my orchard, but not in large numbers. Another one of my goals is because peaches lack hardiness, I've been trying to increase the hardiness within the species by crossbreeding some of the better varieties with a wild relative that has good bud hardiness. So today I'm going to summarize some of the things I've found out about peaches and some of the successes I've had with the breeding. But before I move on, I'd like to acknowledge the research team in Maine and New Hampshire. And also some of the funding sources, the block grant from the state of Maine and growers in Massachusetts. Now, I've talked about this in some of the presentations in the past, the different strategies for reducing winter injury. One of which, of course, is selecting hardy varieties. But the other one is to plant your peach orchards at high elevations. I've seen peaches at higher elevations where air drainage is good have better survival than orchards located at lower elevations where we get when we get the inversion freezes where the warm air is actually at higher elevations than the colder spots because of the elevations that occur when there's no wind in the early hours of the morning. There are two types of freezes that cause winter injury, and where we get the inversions of warm and cold air. This would be a radiational freeze. And it's gravity that pulls the cold air down into the low spots and allows the warm air to rise to the higher elevations. And this type of freeze event occurs more often than the advective type of freeze, where a cold air mass will move into the region, usually with strong wind, and then it's the reverse. I see more damage at the higher elevations. But this type of freeze event doesn't happen as often. Returning to the subject of peach varieties, we all know that hardiness varies among the different varieties available and ranges from -4 in the more tender types to -13 in the hardier varieties. Now, I've heard of reports of peach buds being able to tolerate -17 when they're fully acclimated or artificially acclimated in research trials. And I have seen this in some of my peach trees here at Highmore Farm, but it's fairly uncommon. In the winter of 2023, where we had -18, two varieties had a fair amount of bud survival. It was Garnet Beauty and the rootstock Bailey, but more often it's the limit of hardiness is about -13 to -15. And in our part of the region, we more often lose our flower buds in February and January, less often closer to full bloom. Some bud loss can be beneficial to crop load management, but when it's more than 50%, I've noted that it does have an impact on yield. So the other strategy that I want to focus on is selecting varieties with good hardiness. And I'd like to mention a previous research project led by Bill Shane in Michigan, where he measured hardiness on a large number of varieties, some of which I don't have in my collection. And you can find it at this internet address. And I think I'll send this internet address to Liz after I'm done talking today. But I'm focusing on two different collections of varieties, one in New Hampshire, which I've talked about before, and the new one in Maine, which I'm going to focus on today. I measure hardiness two different ways, similar to Jason, but I don't do the electrical conductivity. I use artificial freezing, where I collect shoots and put them in the freezer and expose them to different lethal and non-lethal temperatures, and then I cut the buds and examine the pistils inside the buds, looking for browning or green. And it's the pistil that's the part of the flower that develops into the fruit in peaches. I'll also use test winters, and when it doesn't, this winter was a test winter because the temperature dropped to about -10 here at Highmore Farm, which is in the range where it kills some of the buds, but not all. And I can pick out obvious differences between the varieties. But when it gets as cold as -18 and it kills all the buds, it's not a good test winter because there's no point in comparing the varieties when everything's dead. So these two graphs show the percent flower bud survival in two of the varieties that were in the New Hampshire trial, Scarlet Rose being the hardiest this year and Desiree one of the more tender ones, at the percent survival at the different temperatures. And non-lethal temperatures would be above -4 and super lethal temperatures below -13. But within the critical range of -4 to -13, the differences between the varieties is pretty obvious. And this is one of the ways I use to determine whether the variety is hardy or tender. And then this graph shows the recent results from Maine with PF15A, which was the hardiest this year, and Desiree, again, which was one of the more tender ones. We also like to calculate the lethal temperature for 50% of the flower buds, and we call this the LT50. And we extrapolate between the data points using a mathematical equation to precisely estimate the temperature that causes 50% bud death. We don't know ahead of time what this temperature is going to be, so we use this mathematical equation. And in the case of PF15A, the lethal temperature for 50% was -9, and it was -6 for Desiree. So this is the other way that I also compare the varieties. After 3 years of doing this with 33 varieties in the New Hampshire trial, we came up with these rankings for the different varieties. With the hardy varieties, these varieties were consistently hardy in the 3 years. Several were only intermediate, and most of them turned out to be fairly tender. So these are the varieties that I would suggest you not plant if you actually want to have a decent yield on your peach trees. The inconsistent column, these varieties were hardy one year and tender the next year. And I think it has to do with some of them coming out of dormancy a little too early and losing hardiness, or the fact that they simply like to respond to those warm temperatures and are unable to regain hardiness when the temperature goes back down. So I've written a report summarizing the 3 years that I studied these varieties, and it's going to be published this spring in the Fruit Quarterly of New York and the Fruit Notes of New England. But the full scientific paper was published in the October issue of Hort Science, which you can access through this internet address. So the variety trial in Maine has about 21 varieties, and I was able to measure some of these some years, and others didn't have enough shoots for a full set of measurements, so they got excluded. But I selected these varieties for their winter hardiness and bacteria leaf spot. And the hardiness rankings were based on what I got from the Adams County Nursery Catalog. They're all on a Bailey level and controller rootstocks, and they're planted fairly close. And I did the measurements over the last 2 years. These are the graphs of the lethal temperature last winter in January and this winter in December. And the varieties are sorted from hardy to tender in both years. Last year, the lethal temperature ranged from about -7 to about -13. And this winter, a little bit warmer than less hardiness than last year for reasons that aren't fully known, but I suspect it's because the trees this year actually had a crop of peaches compared to last year when they had no peaches. So let's look at some of the individual varieties. PF15A was consistently hardy both years, as was Messina. Coral Star and Glowing Star were also consistently hardy in the 2 years. And consistently tender in the 2 years were PF23, Summer Serenade, and Desiree in the year that I measured it. Despite last year being a fairly mild winter, I saw some bud death when I measured percent bud survival in April, and it's somewhat correlated with the hardiness I measured in winter, but there were some surprises, notably PF23, the variety that's supposed to be tender, had reasonable bud survival. And Coral Star and Messina, which were supposed to be hardy, did not have good bud survival. So there's some unknowns in attempting to categorize these varieties as being hardy or tender, and it probably has to do with the dynamic nature of hardiness, which does change considerably over the course of the winter. So some of the things I'll be looking at in the coming years would be the chill requirement of these varieties, which is largely unknown, and whether or not they lose, how much hardiness they gain and lose as temperatures fluctuate through the winter. This year has been a test winter. We had -10 thereabouts in January. And of course, I went out and collected shoots and measured bud loss after this. And these are the results of this initial cold snap. And it did follow cold hardiness somewhat, but there were two surprises. Desiree, which has been tender in some of the other research trials, had the best survival after this cold snap. And Messina, which I found to be hardy, lost a lot of buds. So Jason's already explained this somewhat, but this is one of the things that I think is affecting the hardiness of our peaches, the dormancy or the chill requirement, the number of hours they need to be exposed to temperatures in the range of 35 to 54 degrees. And it varies a lot in peach varieties from less than 400 hours to about 1,200 hours. Here in the Northeast, we generally don't grow the peach varieties that are considered short chill. These would be varieties that they grow in Florida. But we do grow some medium chill varieties, and I think these are the ones that are being anomalous in how hardy they are. But the chill requirement is, it helps trees understand where they are within the winter season, and it tells them when it's time to start growing again. But short chill varieties are going to be fooled into thinking that springtime has come, and they're going to be suffer a great deal of bud loss. I would like to point out that freezing temperatures do not count towards the chill requirement. If the temperatures are below 0, the trees are not accumulating any chill. And if the temperature is above 54 in the early part of the season, they could actually reverse. It's called negative chill. So this graph shows the course of accumulating the chill requirement in Maine. Last year, the solid line from fall into March, and this year, the dashed line, and these would be the chill units from 0 to about 1,300. And the low chill varieties, which would be less than 600 hours, usually get their chill requirement by November. And we don't grow these varieties in the Northeast. We do grow some medium chill varieties, which require 600 to 800 hours, PF23 and Glowing Star. And these would be out of their dormant period by the beginning of winter and ready to respond to warm temperatures throughout the winter. High chill requirements, Red Haven and Coral Star will not come out of dormancy until about March in the types of winters that we've had last year and this year. So for a lot of the other varieties in the trial, I just don't know what the chill requirement is. So this is something I'm going to be measuring in the years to come. And hopefully it'll help us identify which varieties we should not be planting. So after 2 years, these are the hardiness rankings I've assigned to the varieties in my trial. The three that were consistently hardy were Glowing Star PF15A and Coral Star, and these would be the tender varieties. Autumn Star, Blushing Star, PF23, and one of my favorites, Summer Serenade. And of course, the big question is, how does this affect yield? It does affect yield to some extent, but there are other factors involved. This would be the yield I measured in these trees last year in the older block and the younger block, sorted according to yield per tree in pounds per tree, with Messina and PF15A out yielding a lot of the other varieties. The yield measurements last year were complicated by the large amount of brown rot, so I wasn't able to harvest all the fruit. So I simply estimated the percent that were lost to brown rot and added that to the harvested yield. And so these represent total yield in these varieties. And so it didn't 100% correlate with bud hardiness, and that's because some of these varieties don't produce a lot of flower buds. So they may have 100% bud survival, but if they only produce 2 or 3 buds per shoot, they're going to have less yield than something that produces a lot of flower buds but has 50% bud kill. So it's the combination of these two that contributes to yield. So the other side of hardiness would be tree hardiness, and I don't do as much measuring and observations of this. But in the New Hampshire trial, I went back in May of 2024 and looked at each and every tree and assessed them for how well they were doing. And Scarlet Rose was doing the worst. It did have one of the better levels of hardiness in the flower buds, but I'm not recommending it because the trees tend to die fairly quickly. So, and they also get cankers. These branch junctions are more tender than the rest of the tree, and this part of the tree gets injured first. And the cambial tissue, it's in between the wood and the bark, and it's fairly tender in peach trees. I suspect these cankers occur where the cambium dies, and disease can get in. This is one of the factors of tree hardiness that I think is important. When I measure tree hardiness, I'm measuring the xylem or the wood, and when I see a lot of xylem damage, I see small fruit size. When I see injury in the cambium, I see shoot death and cankers along the trunk. So this would be the summary of the tree health rankings that I did in the New Hampshire trial. So varieties to avoid would be these over here: Sugar Giants, Scarlet Rose, Desiree Galaxy, and PF5D. A lot of the trees are doing quite well, and a lot of them were intermediate, but doing good enough. And this is also in the 2 reports that are coming out this spring. So I'll finish up by talking about some of the breeding success I've had. Because the beech varieties that we grow aren't hardy enough for Maine, I've started breeding new peach varieties using the Bailey rootstock, which has some wild traits. It's got small fruit size, but good bud hardiness, and the fruits don't taste that good. They also have white flesh. And I've crossed them with Hero Beauty and a few other varieties that have very good flavor, but not the best hardiness. And at this point, I only have 127 new seedlings in the orchard from these crosses, but last year, 7 of them produced fruit and allowed me to measure their hardiness and evaluate their quality. These 7 trees did have small fruit size and pale yellow flesh. They ranged in fruit quality from very good to poor. So they're not ready for commercial production, but I can use them to breed new varieties by back crossing them, and hopefully they'll retain their hardiness. I was pleased with the amount of hardiness that they've had. These graphs show the flower bud survival after last year and after the test winter this year, and they have done, I've called them HF1 through HF7. and their bud survival was as good or better than the hardiest varieties in my trial. So this is just a summary of all of the observations I've had from the variety trial in Maine with the type of flesh, the hardiness, the fruit quality, fruit size, and harvest date. And I hope to publish this at some point in the future, but this can be used as a reference to pick out which varieties you think would be good in your orchard.